All right, we're about to jump into something truly fundamental to all life, and then see how the absolutely bizarre rules of quantum physics are helping us learn how to fix it when it breaks. I want you to imagine something for a second. Picture a stack of paper that's 18 miles high. I mean, it's literally reaching up into the stratosphere. Your job is to copy every single page, and in that entire massive stack, you only make one single typo. Sounds absolutely impossible, doesn't it? Well, believe it or not, that incredible, almost unbelievable level of accuracy, that's exactly what's happening inside you right now, every single second. We're talking about DNA replication, and its precision is just nothing short of a miracle. So here's what we're going to cover. We'll look at how this perfect copy machine works, what happens when it breaks, and this is the really cool part, how digging into its quantum mechanics is paving the way for some incredible new medicines. All right, let's do this. Okay, first things first. We really need to try and get our heads around the sheer scale of this. I mean, what our cells are pulling off every single second of every day is just mind-blowing. So look at that number, 1 in 10 billion. That's the final error rate after all the proofreading and quality control is done. One mistake for every 10 billion letters it copies. To give you some perspective, that's like typing out the entire Encyclopedia Britannica more than 200 times and making only one single typo in the whole lot. It is just ridiculously precise. And how does this all get done? Well, it's a whole team of tiny molecular machines. You can really picture it like a super efficient construction crew. First up, you got helicase. That's the one that unzips the double helix. Then primase comes in and lays down the starting blocks. After that, you got the superstar, DNA polymerase, which just flies along, building the new strand at an incredible speed. And finally, ligase comes in to seal all the little gaps. It's this beautiful, coordinated dance, and it's happening in trillions of your cells as we speak. But, when you're making billions and billions of copies, even a system that's almost perfect is going to make a few mistakes. And when that happens, well, the consequences can be devastating. So one of the main villains in this story is something called replication stress. The best way to think about it is like a huge traffic jam on the DNA highway. Anytime that copying process gets slowed down, jammed up, or disrupted, that's when errors really start to creep into the code. And here's the critical takeaway. These aren't just harmless typos. These errors can make the whole genome unstable, what scientists call genetic instability. And that instability, it's a direct road to mutations, and is one of the fundamental causes of cancer. And this isn't some abstract textbook concept. You probably heard of the genes BRCA1 and BRCA2, right? Well, their whole job is to be part of this DNA repair crew. So what happens if those repaired genes themselves are faulty? The person's risk of developing certain cancers just skyrockets. It really drives home how absolutely vital these repair systems are for keeping us healthy. Okay, so that raises a big question. If the initial copying process makes mistakes, how on earth do we get to that insane 1 in 10 billion accuracy rate? Well, it's because there's a quality control team that comes in right after. It's called the mismatch repair system. And just look at the difference this system makes. It is staggering. Without it, you'd get about one error every 10 million bases, which is still pretty good, honestly. But then the mismatch repair system kicks in, and boom, the accuracy jumps by 1,000 times. 1,000. It's the ultimate proofreader. Which brings us to the central mystery, right? How does a tiny molecule know that one chemical arrangement is perfect, but another one, which is almost identical, is a dangerous mistake? Well, the answer isn't found in classical biology. For this, we have to go quantum. Yeah, so scientists now believe this isn't a simple mechanical check, like a key fitting into a lock. Nope, it's something way more sophisticated. They're proposing it's a full-blown quantum recognition system. The whole process is called active recognition. And that word active is, well, it's key. The enzyme isn't just passively bumping into the DNA to see if it fits correctly. It's using energy from a molecule called ATP, which is basically the battery pack of the cell, to power up and actively interrogate each base pair. This is what allows it to be both incredibly fast and unbelievably accurate. So here's the simple 3-step breakdown. First, our little enzyme powers up using that ATP energy. Second, it physically interacts with the DNA, and for a split second, they become a single, connected quantum system. And third, that system has to relax back down to a low-energy state. The specific path it chooses to take, well, that's the decision. And this is where it gets so incredibly cool. You can think of it as a quantum lever. For a correct match, the path of least resistance, the easiest energy pathway for the system, is the one that basically says, all good here, move on. But if there's a mismatch, that one wrong piece completely changes the quantum physics of the interaction. The whole energy landscape flips. Now the easiest, lowest energy path is the one that says, whoa, error detected, sound the alarm. The physics itself forces the right decision. It is just brilliant. The researchers even compared this enzyme to a famous old thought experiment in physics called Maxwell's demon. The idea was about a tiny being that could sort fast and slow particles, creating order out of chaos. And in a very real way, that's what this enzyme is doing. It's a real-life Maxwell's demon, using energy to sort correct from incorrect and maintain the perfect order of our genetic code. Okay, so quantum mechanics, Maxwell's demon, this is all fascinating stuff. But what's the real-world payoff? Why does this matter to you and me? Well, it's simple. This fundamental understanding lets us design much, much smarter medicines. And the logic behind it is just so elegant. Step 1, you map out the normal machinery. You have to understand how it works perfectly. Step 2, you find the exact part that's broken in a particular cancer cell. And then, step 3, you design a drug that is tailor-made to target only that broken part. This is the whole idea behind precision medicine. Kill the cancer cells, but leave the healthy ones alone. And I gotta stress, this is not science fiction. These are real drugs being used in clinics today. Take PARP inhibitors. They are designed to be especially deadly to cancers that have those faulty BRCA genes we talked about. See, those cancer cells already have a weak repair system. This drug comes in and takes out their backup system. With no way left to fix its DNA, the cancer cell just self-destructs. We're exploiting the cancer's specific weakness against it.
So what's on the horizon? Where does all this incredible research take us next?
Well, the truth is, the complexity of these systems is just immense, way beyond what any single human mind can fully grasp. The next wave of breakthroughs is going to come from using artificial intelligence and powerful supercomputers to model these quantum interactions. That's how we're going to find new drug targets and design medicines with a precision we can barely even imagine right now. And that leaves us with a pretty profound final thought. For centuries, humanity has been trying to just read the blueprints of life. But by understanding it down to this fundamental quantum level, we're moving beyond that. We're learning not just to read the code, but how to debug it and maybe even rewrite it. The big question for our future is no longer just what does the code say, but rather, what do we want it to say?